In a groundbreaking study published on July 26 in Nature Genetics, the Lymphoid Development Group at the MRC Laboratory of Medical Sciences unveiled a remarkable discovery. They found that the contribution of cells expressing maternal or paternal X chromosomes can be selectively skewed in different parts of the body. This revelation was made possible by leveraging human data from the 1000 Genomes Project, combined with mouse models of human X chromosome-linked DNA sequence variation. Until now, it was believed that the usage of maternal and paternal X chromosomes was consistent throughout the body. This new research, however, demonstrates that this is not the case. Different organs may favor either the maternal or paternal X chromosome, driven by competition between cells expressing one or the other. In some individuals, heart cells predominantly use the X chromosome from one parent, while immune cells almost exclusively rely on the X chromosome from the other parent. Biological females inherit two X chromosomes, one from each parent, along with all the other genetic material that builds and sustains the body. Despite the presence of both parental X chromosomes, only one X chromosome is actively expressed in any given cell. This selective expression means that each cell effectively chooses a set of unique characteristics derived from either one or the other parent, adding a new layer of complexity to our understanding of genetic expression. We realized that when cells chose one of their two X chromosomes over the other, they also chose which set of genetic variants to express, explained Matthias Merkenschlager, head of the Lymphocyte Development Research Group. This insight opens new avenues for exploring how X-linked genetic variants shape organismal development and the potential impact of selective X chromosome usage on certain conditions later in life. The researchers focused on a specific gene on the X chromosome, known as STAG2. They discovered that cells with a genetic variant of STAG2 failed to develop into lymphocytes in females carrying the variant on one X chromosome and the common version on the other. Conversely, cells with the same variant of STAG2 were fully competent to form lymphocytes in XY males or females with the variant on both X chromosomes. This led to the conclusion that the presence of cells expressing the reference version of STAG2 prevents variant cells from forming lymphocytes, highlighting a competitive interaction between cells. These findings reveal a new dimension of X-linked diversity, where cell interactions shape the contribution of genetic diversity to specific cell types and tissues. Even if cells expressing the reference STAG2 outcompete others in forming blood cells, cells expressing the variant may predominate in other parts of the body. This discovery sparked a personal curiosity for the study's lead author, Teresa Buenaventura. Working on this project has been particularly exciting for me since it has made me curious about the contribution of each of the X chromosomes to my different tissues, she shared. This research uncovers a previously underappreciated aspect of X-linked diversity, where interactions between epigenetically diverse clones shape the genetic diversity of specific cell types and tissues.